Dan the man, long time no
Donny,
see, long time no.
It's been a while. It's been some time.
It's been some time. I hope you've missed me as much as I've missed you. How you been?
You know, my heart aches for how long it's been. Let me tell you.
Well, that's why I joined. I sense the sadness.
No,
in
let's
you, Dan,
talk about Chromosome 2. What you got? Bring it.
Well, some interesting points, some good points, especially with the confirmed prediction aspect of the fusion. And so where I could start, I think, is if we were to assume, for sake of argument, Praise, if you could share my screen real quick.
Oh, here we go, dude. It's already midnight for me. I don't have time for all the slides. Just say it. Just use
We'll
words.
be quick. We'll be quick.
Oh, it better be,
but
man.
This stuff is technical.
No,
it's supposed to be.
Donny, can I just say one time, we got to chat about like graphic design, because this blue background with the black text and the tiny little font, it causes me physical pain.
It's just more evidence of genomic degeneration. Dan, you messaged me what color you prefer. And for our next talk, when you have more time, we could do it on your channel.
Yeah, that'll be 2020.
I will personally alter the color of my slides to your liking. So I would say, even if there was a fusion event, I'm not totally against there being a fusion event, because we can go slide by slide, point by point. I think there's strong evidence that there's not a fusion event. Dan, you pointed out that you believe it's a weak line of argumentation to say that the fusion site itself represents a functional component of the DDX11L2 gene. We can touch on that a little bit.
It's a pseudogene. It's a pseudogene. It's not a gene.
Well, I definitely have some thoughts on that, but let's start here. So even if there was a fusion, I'm not totally against it, because as you can see here on the slide, it would be an apomorphy, since it is a trait observed in a single lineage. So basically the fusion, correct me if I'm wrong, Dan, from your model, from your position, the fusion would have taken place in the human line sometime after the split, right? Humans and chimpanzees go back to a common ancestor 6 to 10 million years ago. At some point in the human lineage, there would have been a fusion event. Is that right?
Yes. So on the hominin side of that divergence, there would have been a fusion on the human lineage side, not the chimp lineage side. It is late, so please forgive me for asking, or maybe requesting. Can you be quick to get to the point here?
Well, that would be my point. You just agree.
Point is what? That it's an apomorphy? That's just a derived trait. Like, what's the... Yes, it's derived specifically in the hominin lineage.
Right.
Okay.
And so if there was a fusion, the biblical model, we have what? We have a flood bottleneck where you have 8 people. We actually have another bottleneck with the Tower of Babel. And so there could have easily been a fusion within that time period where genetic drift can allow for this discrepancy, this change in the genome to become fixed. So either way, I would say it's agnostic to the debate of creation versus evolution, since, as you've admitted, it is something that would have happened in the human line. And so that could either be evidence for evolution or creation. Go ahead.
Can I make sure I understand what you're saying? So you're saying that humans would have been created with 48 chromosomes like the other great apes, and then at some point, due to whatever genetic factors, the fall, small population size, drift, bottlenecks, whatever, that this fusion could have occurred within the young Earth human lineage, and all extant Homo sapiens now have 46 due to this end-to-end fusion. Is that that's the possibility that you're proposing here, right?
That is a possibility. Okay. Humans.
So, yeah, got it. So here's why that's wrong. So not to put too fine a point on it, because what I'm saying is that this is a specific prediction of evolutionary theory, right? Human chimp common ancestry. We can observe the different numbers of chromosomes, the different genome structures, the banding patterns in the different chromosomes, and we make a specific prediction that we should find a unrepresented chromosome in Homo sapiens and 2 unrepresented chromosomes, right, that don't have a counterpart in the chimp genome. And those things, when you line them up, should have a certain set of features. They should have sequence homology. They should have end-to-end telomeres. There should be a vestigial centromere in human chromosome 2, right? Those are specific predictions that must be the case if the evolution model is correct. Now, you're saying that the creation model can accommodate those observations, the observations that we find that those predictions are accurate. And that's true. The creation model can accommodate those observations, but that's a different thing from saying that they are necessary predictions to be
Dan, let me address those points
with the creation model.
Okay, I appreciate that. I also heard some background noise, whoever that's from. Since it's Dan and I interacting, please mute. Okay, so I would argue, going back to your admission, obviously this would have been a fusion event that occurred in the human line. When we actually
it's an admission, that's like the point.
But well, here's the thing: you want to use this as evidence for
a relationship between
humans and chimpanzees.
Yes, because it's a confirmed prediction. Yes.
Now you're saying it's a confirmed prediction, and I am not necessarily arguing against that claim. But what paper, what year, who made the prediction? How was it predicted? What was said? Was it super specific? Was it a little vague? Do you have the answer to that?
I do about an hour-long human evolution lecture every summer, and you're welcome to come on sometime and we can walk through it. But I mean, suffice it to say, yes, this was incredibly specific prediction.
Who made the prediction? What's the name of?
I don't remember the specific paper. It's been a while. Like this, and when I say it's been a while, I mean this was something from decades ago. This was a long time ago.
So
this
they
was a very specific
genome.
human prediction.
Okay, so
it was from before sequencing. It was from before sequencing. The prediction was from before sequencing.
Okay, so you're saying before we knew that humans have 46 chromosomes and the great apes have 48, they made this prediction and it was specific.
So the prediction is based on the fact. So did you catch my conversation earlier with Jamie? Because what it was is go back over 100 years, go back to the 1800s and we say human chimp common ancestry. Then you get to the early 1900s. And that's where we figure out chromosomes are a thing for heredity. It wasn't until the early 1900s that people knew that chromosomes mattered for heredity. Right. So you get to the early 1900s, and now people are looking at different genomes, and you could stain the cells and count the chromosomes. So it's somewhere between, I don't know specifically when, but it was somewhere between 1903 and 1940, probably, give or take, that we realized chimps are 48, humans are 46. It's probably somewhere in that range. And so now we can make specific predictions, right?
Okay, so it was predicted. Wait. So it was predicted after the knowledge was accumulated that humans have 46. Okay.
Yeah.
And that's why I just wanted an answer to that. I don't want to spend too much time on one point because I know you don't have a lot of time. I would argue actually when you do compare chromosomes in humans and in chimpanzees, I don't want to spend too much time on this because I do want to argue for the functional nature of the so-called fusion site, which I know you disagree with the most. But I would argue, for example, I'm sure you're familiar with the Ventura paper. I know you are. I've seen you guys critique it before. And so there are aspects of human chromosomes and your great ape chromosomes, specifically chimpanzee and gorilla chromosomes, that do have some pretty significant differences. Here in this paper, it talks about the presence of these large blocks of subterminal heterochromatin and this lack of satellite DNA around human chromosomes.
Yeah. So what's heterochromatin specifically?
It's repeated DNA basically.
Not necessarily. It doesn't have to be repeated, but structurally, what's heterochromatin?
Structurally, what is heterochromatin? Basically,
what is heterochromatin?
Well, it's basically pieces of DNA within the chromosomes that is repeated oftentimes. That's my understanding.
It's not. It doesn't have to be repeated though. So the characterization heterochromatin is independent of the sequence. It refers to the physical conformation of the DNA.
Okay.
What does that mean? What is heterochromatin?
Well, I think you're playing word games here because it is. Wait, Dan. I want you to answer the question: why is this DNA? Well, why is this structure within the DNA? These structures within the DNA.
I'm answering. I'm in the process of answering that question, but I think you're making the argument without knowing what the words mean. Do you know what
how would you
heterochromatin
define
is
it if it's not just repeated pieces of DNA basically around the chromosomes? That's my understanding. So if you want to,
okay, so you're wrong. Okay, so heterochromatin refers structurally to DNA that is in a condensed state in the interphase nucleus, right? So do you know what those words mean?
In the
condensed, so like structurally it is supercoiled rather than being in a diffuse state, right? So the nucleosomes are not exposed, right? They're supercoiled so that you don't have like access to enzymes that would do things like express genes. Okay, that's what heterochromatin is.
Well, it's thought to be tandem repeats.
It is largely repeats, but it doesn't have to be because it's cell
here's
type
the thing.
specific.
Listen, here's the thing. You just want to posture because I did give the correct answer, and then you want to get hyper technical, which I'm fine with.
You didn't give the correct answer.
No, I gave the. I've seen you all talk about heterochromatin before. I've studied heterochromatin. It's primarily thought to be tandem repeats, and it's condensed pieces of DNA known as satellite DNA. Okay, fine. Answer the question. How come the presence of this is lacking around human chromosomes, where it shouldn't be lacking if we do share a relationship, or specifically if there was a chromosome 2 fusion?
So if you have highly condensed DNA, it's not being expressed. It's not being used. So that DNA is not under purifying selection. So deletions or translocations or rearrangements in those regions are not selected against. That's why. So you tend to have structural differences in regions where you have heterochromatin in one genome, but it's missing in closely related genomes. Because
is it missing because it was lost over time? It was lost over time?
Deletions are relatively common. If they affect a gene, then that tends to be highly deleterious. But if it affects exclusively heterochromatin,
so at what point in the split between the humans and chimpanzees' common ancestor was all of this satellite DNA, this specific kind of DNA, lost? At what point was it lost?
So we can't actually say that it was gained or lost specifically if we're only looking at chimps and humans. And I'm not familiar with the specific regions that you're talking about. Are you talking about regions in chromosome 2 that are shared across humans, chimps, gorillas, orangutans, and other apes? Or are you talking specifically about chimps and humans? Because that matters for the question of when was it lost.
I do believe it's species specific, but specific to chimpanzees, gorillas, and humans. The major differences are with those three. If it's chimps, gorillas, and humans, then what probably happened is that it was a deletion that affected the hominin lineage, but not the other two. And the hominin lineage, you're saying?
Hominin, so the
hominin,
human,
right? So the human line,
human side of the human chimp divergence, and so because it's heterochromatin.
I just want to ask you: is the function of the heterochromatin to make the DNA more condensed, like you were saying? Is that the function of it?
So the point is that heterochromatin is largely non-functional DNA, which, if I recall, I believe you don't think exists very much. But so the point of heterochromatin is that it's persistently condensed DNA.
Okay.
That is not doing a whole bunch of anything. So basically, they put it.
From my understanding, that is wrong, because I could go through papers where this kind of DNA does.
I'm just going to refer you to my chat with Fuzzrana from last year on that one. But not to get sidetracked on it. But so
would you deny the fact that there are papers going over the functional aspects?
Look, I don't want to get sidetracked from the human. I don't want to get sidetracked from the human chimp thing. So
let me just respond to what you were saying. Then I noticed that you said, well, probably there is a deletion, and see what this is a lot of evolutionary philosophy coming from you, because you were saying we have this perfect match, exactly predicted humans and chimpanzees around these chromosomes, specifically
referring to specific regions.
But I am showing you that we have a specific kind of DNA that even those on your side are recognizing is a discrepancy. It lacks in human chromosomes, but it exists. It's in abundant, from my understanding, in chimpanzee and gorilla chromosomes. You're saying, well, probably some kind of deletion or something happened through purifying selection in the hominin line. Well, that's nice, Dan.
I didn't say it was storytelling.
It's a lot of evolutionary philosophy.
With Stan, I'm not saying anything was selected here. I'm saying it was neutral. It's stuff that you don't need. So that's the
no, but that's where your evolutionary philosophy
I don't know where
comes
you're
into
getting
play. Okay,
selection.
I'm looking at a paper right now. It's called Functional Significance of Satellite DNAs, Insights from Drosophila. So there are papers going over the functional aspects of satellite DNA and
no, I know they exist.
Well, okay,
I know
okay. I know you don't have a lot of time, so I do want to move on to. I would argue. Okay,
hang on.
Go ahead.
Can I get a word in here, because I feel like you're jumping around and you're ignoring the actual point that I came in to make.
Well, it's because you're saying, Dan, you're saying more things wrong in one sentence than words that are in that sentence. You're saying a lot of storytelling on how this was lost. You're saying, well, it's just non-functional junk, and so it can be deleted or lost over time. That's wrong. That's false. It's not just non-functional junk. You're saying that, well, the DDX11L2 gene is a pseudogene. Again, that's evolutionary philosophy. Okay, go ahead.
So I would like everyone to pay attention here to what Standing is doing. I came in to answer a very specific question from Jamie, and I saw people were giving Jamie crap in the chat about this. I don't know why people are doing that. That was nice. Jamie, thank you. I think we had a nice conversation. Now, I brought up a couple very specific points in terms of the structure of the chimp chromosomes and a couple very specific features of human chromosome 2. And now Standing comes in and is talking about all these other things that aren't related to what I brought up.
It is related to the chromosome 2 fusion.
Please don't interrupt me. Please don't interrupt me. I am happy to have a conversation if you want to set up a time and we can talk about all this other stuff that you seem to want to bring up. But that's not what I came in here to talk about specifically.
It's all the same topic. It's all the same topic, Dan, and you're the one that started interrupting when this discussion started. I barely got 3 words in before you interrupted.
Fusion site? Do you have anything to say about the fusion site or the pseudogene, the head-to-head telomeres, the vestigial centromere? Do you have anything to say about that stuff?
Okay.
The confirmed predictions.
I just want to sum up this argument for the audience so they can see what we're talking about. I came in and told you, for sake of argument, if there was a fusion, I don't think it's a problem for the creation model because it is an apomorphy. It would have occurred in the human line.
What does apomorphy mean?
Well, if you look at the screen, a novel evolutionary trait.
Yeah, it's a derived trait, right?
To a particular so it's unique to the human line. But we can. But then you said, well, it doesn't matter because when we look at human chromosomes and chimpanzee chromosomes, it matches predictions and expectations. And I showed you one paper that says, no, it doesn't because of this lack of satellite DNA.
You're talking about two different things.
I do want to move on.
No. We're not going to move on from that because you're conflating 2 different things. The predictions that I'm talking about have nothing to do with satellite DNA. Well, you didn't even specify that satellite DNA was in
You're misrepresenting my position. Wait, you're misrepresenting my argument. I am saying it could have happened in the human line.
I agree.
Genetic drift, bottleneck, sometime after
the
flood.
It could have, but it's not a specific required prediction of your hypothesis. It is a specific required prediction for the evolutionary hypothesis, and it's a confirmed prediction.
No, but I would say there's a lack.
It's not equivocal.
I would say there's a lack of consistency with the evolutionist explanation because of the lack of satellite DNA. This very specific kind of DNA.
That is a different thing.
I would disagree with that. I would disagree with that. Okay, so we can move on, though, and we can agree to disagree, okay? So you're trying to say that it's a pseudogene. Well, firstly, anytime you see pseudogene, especially for the audience sake, that is the evolutionary assumptions, just like we saw with the satellite DNA.
No, it's functional. It's been excessed.
Dan, you have to admit, you're interrupting me, too. There's healthy interruptions. You can have as much time as you want to respond, okay? I understand you're going to disagree with a lot that I have to say. I disagree with a lot of what you have to say. I'll try not to interrupt anymore. You try not to interrupt anymore, okay? We understand that many pseudogenes are now necessary to sustain healthy life processes in the cell. I understand you disagree with that, and I appreciate you not interrupting, okay? So what we have here, and Dr. Jeffrey Tompkins, who I know you love so much, he says the purported fusion site is a functional DNA binding domain inside the first intron of the DDX11L2 or 11L2 non-coding RNA helicase gene. So he's saying basically this so-called fusion site, which is about 800 base pairs.
That word, was it non-coding?
What does that mean? Non-coding DNA
what does that mean?
A lot of necessary essential functions. And actually, non-coding DNA is not coding DNA, so it's not.
But you're saying it's not a pseudogene. What does non-coding mean?
When you say pseudogene, you're trying to say, well, this is a genetic mistake, a broken gene, an evolutionary leftover.
What's the definition of pseudogene? What's the definition of pseudogene? Do you know?
A pseudogene is a broken protein coding gene.
Right. And it's broken, so it's
but you're assuming that it's broken. You're assuming that it's broken. I am saying that we know regulatory regions can be non-coding, and so you're looking at this and you're assuming that it is a broken protein coding gene. I am saying no, it's not a broken protein coding gene. It's a no. Wait. A lot of the beginnings of your sentences start in the middle of mine. I am saying that this non-coding RNA helicase gene, okay, the DDX11L2 gene that overlaps the so-called fusion site. The so-called fusion site is acting as a second promoter for that gene that is not a broken gene. And here's the last thing I want to say is, how do you get highly essential and functional genes by slamming together two chromosomes? Go ahead, take your time.
So here's the thing. One, we know this is not a sequence. This region is not under purifying selection. The promoter that Tompkins is pointing out here, promoters are, does everyone know what a promoter is? Does everyone know what a promoter is? Standing, why don't you explain to everybody what a promoter is?
A promoter initiates transcription.
A promoter is a transcription initiation site. They are highly sequence specific. So in order for a promoter to work, it has to be sequence constrained. It has to be under purifying selection. Functional promoters, if they mutate, are selected against, right? The mutated non-functional ones are selected against. This region, this fusion site, is highly degraded and it's not experiencing purifying selection. So it's not a functional promoter. What Tompkins is saying is, you look at this
what makes you think it's highly degraded? Unless you want to just take your time and I can respond.
I'm getting there.
Take your time.
So what Tompkins' argument is that you've got this pseudogene in the subtelomeric region, and there's an alternate start site on the far side of it. So the fusion site itself occurs within an intron of this pseudogene. Now, he calls it, in the quote on screen right now, a functional DNA binding domain inside the first intron of the non-coding RNA helicase gene. Right. It's okay. So that's a little bit misleading from Tompkins there, because there are protein encoding genes, and then there are RNA genes, right? There are genes that make RNAs. The RNAs do something. But a helicase is a type of protein, right? It's an enzyme that does a thing during DNA replication. If it is a pseudogene that is non-coding, it's not making a helicase, because a helicase is a polypeptide, right? And this sequence does not ever result in a polypeptide. So just right off the bat, what Tompkins is saying here is wrong. Like, he is conflating things that are not the same thing right there, right? What you had on screen.
Are you saying that he's just lying? He's wrong about this quote?
I don't want to say whether he's lying or whether he doesn't realize he's wrong, but it's not a protein coding gene, and it's a pseudogene for a helicase. It's a pseudogene.
Nobody's saying he's not saying it's a protein coding gene. And he points out here that the fusion site sequence binds to at least 12 different transcription factors, including RNA polymerase 2, a key enzyme that transcribes genes. Are you denying that? Are you saying that he's wrong about that?
No, RNA polymerases love DNA. They grab on all over the place. They are huge fans of DNA, and it's not hard to get RNA polymerases to bind to DNA. It's very easy to do. You can randomize promoter sequences and you still get transcription. You can literally just take 100 random nucleotides and you still get transcriptional activity. That's been experimentally shown. So, like, you got to show me if you want to show me this is a functional gene, you got to show me transcription, you got to show me translation, you got to show me purifying selection.
Okay, well, here he says,
most you get out of this is transcription.
Well, there's a lot. I mean, I've got pages and pages of notes on.
Is there in all those pages, is there translation and purifying selection? I don't think so.
Well, why would they be so we have DNA to RNA to protein, transcription to translation, translating for a protein. Why would there be translating? It's non-coding. It's a
pseudo
non-coding RNA gene.
Great. So there's no translation. Is there purifying
why
selection?
Would there be translations? It's an active promoter. Notice here the DDX 11L2 long non-coding RNA gene.
I'm not saying.
I just want to finish my sentence, Dad.
I'm not disagreeing.
I didn't interrupt you. Okay, this gene, long non-coding RNA gene, you said it's not functional at all. You said it's a pseudogene, it's a broken protein coding gene. Well, notice this RNA gene containing.
It's not protein coding.
it's a broken which makes it non-coding, and it's degenerative coding.
It's no longer protein coding.
It's no longer protein coding, but at one point it was. RNA gene containing the alleged fusion sequence encodes several transcript variants expressed in at least 255 different cell or tissue types. I mean, that's pretty significant, is it not, Dan?
Honestly, no. To be frank, no.
It's doing something. It's doing something. No?
Transcription. I don't know if people appreciate just how messy transcription is. I don't know if that's like a common thing that people think about, is that transcription is a noisy, messy process. And even when it's highly regulated, which it largely is, there's still a lot of messiness in terms of what enzymes can grab onto.
It sounds like you're assuming it's messy. It seems like more.
It's not an assumption. It's been evolutionary documented that it's messy. This is not an assumption, sir.
I mean, does it not require a lot of energy within the cell to add a nucleotide, a single nucleotide to
adding a single nucleotide requires 1 ATP or equivalent? Yes.
It still requires ATP synthase. That's 1 nucleotide to a growing RNA chain. I'm just seeing a lot of evolutionary philosophy, a lot of evolutionary base assumptions with very little evidence. Again, we see that it's doing something. We see that the site, as you've admitted, is very degenerate. It's only about 800 sequences long. Wait, I'm not done.
Or is it functional?
Go ahead.
Is it degenerate or is it
because the so-called fusion site is a promoter for this long non-coding RNA helicase gene that actually overlaps it. It overlaps the fusion site.
Yeah, Tompkins' argument is that the fusion site that the promoter is on the far side of it, that the first exon is on the far side from the rest of the pseudogene. That's Tompkins' specific thing, is that you have a start site that spans it, right? And that the fusion site occurs within an intron.
Right. Yeah, inside the first intron of the DDX. 11L2 non-coding RNA helicase gene. And so it does. It spans the fusion site. And so and you admitted earlier with Jamie that yes, if this was a functional component of the gene that spans it, then that would be good evidence. Sure, maybe this wasn't the result of a hypothetical fusion at some point in the hominin line. And that's what we're seeing. We are seeing a lot of function associated with it.
We're getting bogged down in the minutia here, and I want to make clear for everybody what the point is before I go to bed, because I am going to go to bed in a couple of minutes. But I want to make very clear what the point is here. When we first took a look at the human and chimp genomes, what people expected to see was the same number of chromosomes. That was the original idea, right? That was the original prediction that,
and it failed. It was a failed prediction.
And it was wrong, right? That was an incorrect prediction. So what we said is, okay, if it's the case that humans and chimps share common ancestry, then there has to have been a fusion in the human side of that lineage descended from our common ancestor with chimps. Right, there must have been a chromosomal fusion.
But there wasn't.
If that is the case, if that is the case, this is the confirmed prediction. If that is the case, then we need to find certain features. We need to find an end-to-end telomere in the middle. We need to find a vestigial centromere somewhere in the middle, and
you don't find any of the two.
And these features need to match up in the human chromosome and the 2 chimp chromosomes. Now, when you sequence those chromosomes, you find the banding patterns. Hang on one second, and I'll show you a picture, because you're saying they don't match up, and I'm going to find a picture.
Well, firstly, we've already shown many discrepancies.
Oh, hang on. No. You said they don't match up, and I'm going to find a picture.
Yeah, but here's the thing. You've talked about the telomeric repeats. You've talked about the cryptic centromere. Let's go one at a time, one topic at a time. Notice here on this slide.
Here's the picture I'm looking for. And I'm going to show this one picture because, again, I need to go to bed. I want to address this.
Are you talking about syntony? Wait. Are you talking about syntony?
Hang on. Just give me a moment.
Well, as you're finding it, I want to my screen. If you got to go to bed, then there's a lot of slapping and running here.
Here we go. Here, can I just can someone just put my screen on just for a second? It's literally it'll only take a second to show this, because Standing is saying they don't match, and like this is the banding pattern, so this isn't the sequence similarity. This is just when you stain them and it's looking at different features of the structures. They do match. You can look at this, like they match.
Well,
okay,
you're going to find a lot of similarities, sure, when you're looking
so
at
that's
chromosomes.
okay. Well, that was
a
a
lot of
specific
discrepancies
confirmed.
too.
Well, my point is this was a specific confirmed prediction that the structures are going to match. They're going to have specific features in the same places, which we find. And when you look at the sequences between them, the sequences are going to match. You find the same gene sequences in the same places on human chromosome 2 and the 2 smaller chimp chromosomes. These are all specific confirmed predictions that must be true.
Wait, is this synteny or no?
Well, hang on. Let me finish my point, and then you could say whatever, because I'm going to go to bed. But these are
want
things.
You to stay while I refute your points, though. I want to get a concluding statement, so go ahead, finish, because I don't want you running.
These are confirmed predictions, and again, I'll summarize them, and I'd ask that you just let me summarize it without interrupting. So the size of the chromosomes, the banding pattern of the chromosomes, the structural features like telomeres and centromeres of these chromosomes, and once we had the capacity to sequence them, these specific gene sequences, finding the same genes in the same places, right? So we can line up these chromosomes side by side, and we find a bunch of specific features that are the same between the two, the human chromosome 2 and the 2 smaller chimp chromosomes. My big point here, and the sole reason I joined this stream to just make this point, is that it's not just that we said, oh, there's fewer chromosomes, there must have been fusion somewhere. It's that we have these specific predictions that must be the case if human-chimp common ancestry is true. Those predictions, being accurate, don't invalidate the creation model, but it doesn't matter one way or the other. The creation model can accommodate it either way. But for the evolution model, you must have these similarities between these chromosomes. And it's exactly what we find. They are accurate predictions. And as I said at the beginning, I think before you were here standing, it's not proof by any stretch of the imagination,
right,
that of human chimp common ancestry. Science doesn't deal in proof. Math deals in proofs. Biology doesn't, right? It's not proof, but it's a series of extremely specific predictions that were very specifically confirmed. And that's why it's strong evidence of human chimp common ancestry.
Okay, Dan, I wrote down your points. I am going to address it uninterrupted, and then you know what?
Can I ask a question about that photo?
No. Jamie, it's me and Dan going at it. And then I want to give Dan the last word. Okay, I appreciate.
Well, hold up. I want to take in one question before he goes.
Okay, go.
All right, Mr. Dodgeball Dan.
You know, Bubba, if we're going to get into that, no, I want to finish my thought.
No, if you're going to do that, like I'm sitting here for 45 minutes.
Are you scared of that question, Dodgeball Dan? Are you scared of that question?
Bubblegum, before
we're not going to do
get
this,
lost.
You're scared of that question, or are you?
No. I'm going to finish up with Standing, and if Jamie wants to say anything, I came in to talk to Jamie. Standing joined. We've been having a nice chat. If you're going to come in and insult me, then I'm going to leave. Like, I got better things to do with my life.
Okay, Olive, please mute. Hey, Olive, please mute Bubblegum. I really want to get these points in because there's a lot of points on the table. If you can share my screen, and out of respect, I do appreciate Dan's time here and staying cordial for the most part. I'll give him the last word. Okay, so firstly, yes, they had to invoke a fusion when they realized the chromosome numbers didn't match. Otherwise, evolution fails. And when it comes to the DDX11L2 gene that does span the fusion site, it produces RNA that actually helps regulate the DDX11L gene family, as has been shown with other functional pseudogenes. And so again, the question that I asked, and I don't feel like was answered, but we could have another discussion on this later, is how do you get highly essential and functional genes by slamming together 2 chromosomes? Sounds unrealistic, sounds far-fetched, okay? When it comes to the region, yes, we should have found these telomeres. And what do we find here? Well, Dr. Tompkins, who also addressed this synteny argument in detail in his paper, I actually have the paper printed out. I recommend reading it. And he says actual synteny between human and chimpanzee remains to be resolved until an unbiased assembly of the chimpanzee genome is produced. When you look here, he says an end-to-end fusion as proposed by evolutionists would give a signature of at least 10,000 bases in length, But what we actually find is only 798 bases in length. The evolutionists would just say, well, there's been a split, and so a lot of those bases or sequences have been lost. But it turns out that when you actually do a genome search in all the different chromosomes of these telomeric-like sequences, you find them throughout the entire genome in all chromosomes, with a lot of them associated with significant function, such as gene expression. Okay. And if you read his paper here, Debunking the Debunkers, which I don't see the critics actually addressing, he points out, in summary, chromosome fusions would not be expected to form complexly transcribed and regulated multi-exon alternatively spliced functional gene. This is a functional gene. And in this paper specifically, he refutes Ken Miller's arguments against what he's saying. And these are the arguments basically that are just repeated by people like Dan and other critics. And so I'd highly recommend that they get up to date on the evidence. So no, what they actually find is not what was predicted with the telomeres, with the centromere. Obviously, we see a lack of satellite DNA, and I just don't think it's looking very good for the fusion hypothesis. Last thing I want to say, when you hear the evolutionists like Dan talk about non-functionality or the genome's really messy or pseudogenes, assuming that a lot of these non-coding genes were once functional, unbroken protein coding genes, that is riddled with evolutionary philosophy and evolutionary bias, because we understand a lot of these pseudogenes are necessary to sustain healthy life processes in the cell. And so either way, if there was a fusion, the creation model can explain it. But what the data suggests is there probably wasn't a fusion because of the functional nature of the site itself. I yield, and Dan can have the last word.
Thank you. So I'll just run through a couple points really fast, and then Jamie, if you want to get anything just at the end, feel free. So one thing, the regulating the DDX11 family, I want to just refer everyone to, I chatted with Dr. Fuzz Rana from Reasons to Believe last summer, and we talked about this exact thing for a while. And the idea is that regulation is messy, and the way transcripts interact with, RNA transcripts interact with other RNAs is very messy. And we have very strong evidence that they will interact and interfere with each other, but we don't have strong evidence that it is actually regulation that is selected for. And again, I went into a lot of detail with this with Dr. Rana, and it would be a lot to kind of rehash all of that here, but I'll refer to everybody. You can find my channel, shameless plug. But I chatted with Rana, and we talked about this exact thing for like 20 minutes. Check that out. It was a really good conversation. We go into it. In terms of functional DNA from a fusion, I mean, fusions are really common. So for example, I apologize. I'm going to share my screen just one more time. And it's a figure of chromosomes in two different species of, there are these deer called muntjacs. And, like by any model, evolution, creation, old Earth creation, young Earth creation, by everybody's model, we all agree that these things share common ancestry. They're sister species. The sequences of all of this DNA are almost identical. What's different is the arrangement of the chromosomes. In one species, you have very few, very large chromosomes. In the other species, you have many, very small chromosomes. The way you get from this to this is a whole bunch of chromosomal fusions, just telomere end-to-end fusions, right? So it's not like this is a hard thing to do. This happens all the time in evolution, in places where creationists and evolutionists agree
no, we don't.
that two things share a common ancestry. So that's on the end-to-end, how do you get all this information? Chromosome fusions are easy. It's not hard to do. In terms of the satellite DNA thing, satellite DNA is often species specific, largely because it's not under purifying selection. So what gets retained and what gets lost is there's not expected to be consistency across different lineages if they share a common ancestor with that specific DNA. Why are the telomeres so little? Because they're broken down. They're non-functional. They're not under purifying selection. So any deletions that affect them are not selected against. And then the last thing is just a complex transcribed multi-exon, whatever phrase it was of Tompkins. I want to make sure everyone's clear on this. Transcription is really messy. And transcription costs energy, but so does regulation. Regulation is really, really costly. So you could actually compare the cost of a certain level of transcription versus the cost of completely eliminating that amount of transcription. And what you often find is that the cost of regulating it to zero is more than just letting it happen at a low level. And as evidence that this transcription that we're talking about is spurious is that it has multiple start sites. When you have functional transcripts that are either transcribed or not, it tends to have one start site. Sometimes you get alternate start sites and you get different structures. Largely, you have one start site, and you need that specific thing. When you have multiple start sites that are close to each other, that's an indication that what you're transcribing isn't all that important, because the sequence can vary a lot, and it's not.
Well, Dan, that's kind of a long last word, man. That's not fair.
But the
last word is like sum it up in a minute or two.
I'm going to give you the last word. I'm going to go to bed now, and you can say whatever you want.
Whoa! Don't run away, Dan. I got one question.
No. So stop. I'm not gonna. I'm gonna give him the last word, even though it was a long last word. Closing statement. A lot I want to respond to. I'm not. You're still getting the last word. Share my screen. I'm not even going to say anything or elaborate on this.
You're welcome to do it.
But your point, Dan, on the spurious nature of the DDX11L2 gene, again, it's been addressed. These are repeated talking points. You're getting the last word. I'm not going to go through this slide, but I just recommend people check this out. Debunking the debunkers. It's been addressed. This is just talking points that have already been debunked. So that's all I'll say. Look at that article.
Look, you can say, like I said, you can say whatever you want. I need to go to bed, but the big thing I wanted to emphasize for Jamie was the thing I talked about specific evolutionary predictions. So I appreciate you all letting me come in and giving me so much time. That was a lot of fun. And Jamie and Standing, thanks. I appreciate it. I'll talk to you all later. Bye.
Dr. Dan, appreciate it.